Welcome, everyone, to the latest installment of the Spark Seminar Series. I'm Sue Tappan, a member of the Spark Data and Resource Center. Today's seminar is about one of the most remarkable structures in the human body, the vagus nerve. Running from the brainstem all the way to the abdomen, the vagus nerve serves as the main two-way communication highway between the brain and virtually every visceral organ in the neck, chest, and abdomen. It regulates heart rate, breathing, digestion, and even immune function. It carries sensory fibers reporting back to the brain and motor fibers delivering instructions to the organs, all bundled together in an intricate architecture of fascicles and fibers. That architecture matters enormously. Vagus nerve stimulation has become one of the most promising tools in neuromodulation therapy, with applications ranging from epilepsy and depression to inflammatory disease and cardiac conditions. But we still don't have a clear picture of how fibers are spatially organized within the nerve or which fascicles connect to which organs. Without that, designing precise and targeted therapies is difficult. The NIH Common Fund's SPARC program, Stimulating Peripheral Activity to Relieve Conditions, was created to accelerate the development of therapeutic devices that modulate nerve activity to improve organ function. Operating in two phases and generating over 360 openly available datasets to date. The first phase focused on mapping peripheral nerve topology and studying how nerve activity could be modified to mitigate disease. That work laid a critical foundation, and it set the stage for deeper questions that the second phase was designed to answer. The second phase focused on the vagus nerve, the internal structure, its organ connectivity, and the variability across individuals that has such direct implications for therapy design. It is in that second phase, which is where the REVA project comes in. REVA is the largest coordinated study of vagus nerve anatomy to date. Two research teams came together for this work. One is led by Dr. Stavros Zanos and Dr. Theodore Zanos at the Feinstein Institutes of Medical Research. The other is led by Dr. Andrew Schofstall at Case Western Reserve University and Dr. Nikki Pillow at Duke University. Together, these teams have already contributed datasets to the SPARC portal, representing more than 168 vagus nerves analyzed from 80 human donors, an unprecedented scale for this kind of work. The overarching goal is to create and share a fully quantified map of the human vagus nerve, from brainstem to the abdominal region, capturing fascicular organization, microscopic fiber structure, and organ connectivity across multiple layers of anatomical detail. And all of it will be openly available, which brings me to something I want to say directly to everybody watching today. If you are working on data, models, or devices relevant to the peripheral nervous system, I want to encourage you to explore the SPARC portal. Share what you have. Use what others have shared. This kind of open, cumulative science is how we move faster and how we get better tools to patients sooner. SPARC connects the body and the brain through open resources that support research into systems physiology and neural control in the periphery. That includes our computational platform, OSPARC, and the annotated anatomical maps generated from SCAN, our knowledge base of peripheral connectivity. SPARC is also a member of the AWS Open Data Sponsorship Program, which means all public datasets are stored and accessed without cost to researchers, including you. SPARC is committed to ensuring interoperability across diverse contributions, advancing basic science, clinical applications, and bioelectronic device development together. Today's seminar is actually the first in a two-part series focused on the REVA data. Next week, Todd Levy and Mabel Lin will discuss how individual samples are being analyzed at multiple scales, using computational pipelines to reconstruct and characterize human vagus nerve fascicular networks. That's Wednesday, August 19th at 3 p.m. Eastern. Please mark your calendars because it's the natural continuation of everything you're going to hear today. Now for today's speaker. Please join me in welcoming Dr. Siyar Bahadir from the Feinstein Institutes for Medical Research in Northwell Health. Dr. Bahadir is an Elmezzi Scholar in the Institute of Bioelectronic Medicine, where his work focuses on computational neuroanatomy of the vagus nerve and nodose ganglion, and on bioelectronic medicine more broadly. His research brings together cadaveric dissection, three-dimensional histology, and parametric modeling of the human vagus nerve cytoarchitecture, with the goal of better understanding how the vagus is organized and how that anatomy can inform more selective neuromodulation strategies and improve surgical approaches. Today, he will discuss the Vagus Nerve Explorer and recently submitted work mapping organ-specific branch emergence from the human vagus nerve, outputs made through the REVA project. Please welcome Dr. Bahadir.
Thank you very much, Sue. Thank you very much for the kind introduction. Can you unshare? Thank you.
There you go.
All right.
That
Good
looks great.
afternoon, everyone. My name is Siyar Bahadir. I'm a neurosurgeon and currently a PhD student at Feinstein Institutes at Elmezzi Graduate School of Molecular Medicine at Northwell Health. I'm part of TMP lab. You may have heard my PI's name. I work with Stavros Zanos. And today I want to uh, talk about our recent work on the branching patterns of the human vagus nerve, where we built a standardized map of where its branches leave the main trunk and where where the nerve visibly interfaces with different organs and systems along its course. And if that anatomical separation could help us think differently about selective neuromodulation, and we'll demonstrate a tool we built to explore the anatomy and perform all these analyses that we're going to talk about. First of all, let me start talking about the vagus nerve. Why are we working on vagus nerve? And the vagus nerve links brain and body, as you have beautifully put it. One continuous nerve reaches organs across nearly every major homeostatic system. It protects the airway, it coordinates the swallowing and voice, it senses pressure and chemistry in your major vessels, it participates in cardiac and pulmonary control, so carries visceral signals from the esophagus, abdominal organs. It contributes to digestion, metabolism, inflammation, and other homeostatic reflexes. And even though not every organ is directly innervated by the vagus, very few major homeostatic systems sit completely outside of its influence. So that's why vagus nerve stimulation can affect so many physiological processes, and it is also why it can create effects we did not intended. Uh, and why anatomy? Why are we studying gross anatomy? I'm sorry. Gross anatomy of the vagus nerve. Well, very simple. The neural functions are organized through anatomy, and neural pathways occupy distinct anatomical spaces, and this is valid for everywhere in the human nervous system. This is valid for the brain, this is valid for the spinal cord, and it is obviously valid for the peripheral nerves and the vagus nerve too. So each pathway carries specific functional information, anatomical relationships shape expected functional consequences. And mapping the structure helps anticipate functional change. And this is known for a long time, whether you're a surgeon going in for a tumor or placing a DBS electrode, where you place there, where you stimulate, what happens when you excise some part of the brain is very important. So anatomy is gross anatomy is the most high level view of everything. And why branching? Why do we study the anatomy of branching? We can apply the same logic to a branching nerve. So a branch carries fibers away from the main trunk toward a particular target. Once that branch has left, a device placed below it cannot directly recruit those branch-related fibers through the trunk. So look at the colored pathways in this figure and place the electrode high here. Almost every distal pathway is still inside the nerve. Move it lower. Now you have lost this red branch, that red bunch of axons that has left with this branch. Move it a little bit lower. And then you have lost these, the blue and the purple one, and you're left with this single group of axons that you want to target that goes through this branch. So we wanted to map the branches in this study. We wanted to map the branches. We wanted to standardize the emergence of these uh, branches along the vagus nerve because we're going to talk about why, um, and map it to surgically identifiable landmarks. And then we wanted to quantify patterns of branch emergence. We wanted to estimate the prevalence, emergence patterns, branch-free windows, and differences by site and sex. And then lastly, we wanted to test this postulate. Uh, we wanted to test the separation of these branches based on this postulate to compare target and off-target emergences and examine alternative device levels in terms of possibility of branches to be activated. So I want to first talk about and thank while doing that, talk about our study donors and subjects. So the REVA anatomy teams that were led by Zainab Nasrallah and Mary Barb at Zucker School of Medicine and at Temple University all did all these dissections. It was 28 donors, 56 nerves, and we counted at 2,000 and around 2,200 branches. Um, there are, and all these other people that were also involved. This was the most time-consuming part of this study, and all were done by these amazing people through REVA project. And now let's have a look at the branches. What is a branch? A branch emergence, let's say, is the first visible separation of a branch serving different destinations from the main trunk. So here, a branch to superior cervical ganglion is the first time we observe something coming out of the vagus nerve and joining SCG. And in the case of an aortic nerve, the first visible emergence of something going through the aorta. So gross anatomical dissection allows observations of direct relationships between entire course of human vagus nerve and the organs it innervates. It has its disadvantages. You can't see below some resolution, but it allows you to see the whole forest. And these branches can be thought as the interfaces with organs and systems. They connect to the shared vagal trunk to specific vessels, muscles, plexuses, heart, lungs, and esophagus, and other organs. So, how do we organize this, though? This was great work by Dr. Mary Barb, a very experienced anatomist in peripheral nerve anatomy. So we built a taxonomy of the branches by traced endpoints. So we have 7 main groups and 25 subgroups, and our main groups being sympathetic, muscular, vascular, cardiac, and pulmonary and esophageal. This is the first tier, and it's more a broad organ or systems group. And then there's a second tier of targets, which are actual anatomical endpoints. So sympathetic group has superior cervical ganglion and sympathetic trunk. Muscular has pharyngeal, SLN, ansa cervicalis, generic laryngeal, and recurrent laryngeal branches nerve branches, and vascular has carotid sinus bifurcation, internal or common carotid, internal jugular vein, and aorta branches. Cardiac group has superficial cardiac plexus, generic cardiac branches, deep cardiac plexus, and cardiopulmonary branches. And pulmonary group has only one endpoint, so single subgroup. And esophageal branches has generic esophageal plexus branches, and branches arising from esophageal plexus to the pulmonary and cardiac systems. So these labels, though, they describe trace destinations. Obviously, they are not fiber types. They do not tell us the direction, or and they do not tell us about the physiological effects. We can only infer these through, looking at the organs and what the functions of these organs are in with relation to function of vagus nerve. So let's go over the anatomical examples and how they actually look like, and so we can also appreciate how hard it is to uh, make all these observations and saving them and you know letting us work on this. So sympathetic, and these are here you see sympathetic and vascular branches. So there is a natural progression of these branches, as you are going to see, that from the top of the nerve to the bottom, branches follow very simply the organs or the systems that are next to them. So when a target system is above the other target system, usually the branches also arise above the other one's branches. So here you can see the branches to superior cervical ganglion and sympathetic trunk. These would be our sympathetic group. The vagus nerve has little branches going to this SEG. In this example, can be different in others. And here you can see the B1 is showing a branch to carotid sinus. Uh, Again, the branches to other parts of the carotid bifurcation than the sinus. And then B3 shows again the branches to carotid bifurcation and branches to common carotid artery with relation to it. Common carotid artery, as you know, comes as a single trunk and branches into two at the bifurcation. So bifurcation is higher and the main trunk of the carotid artery is lower. So expectedly, you see the branches to bifurcation to be higher than this. And again, you see another group of branches to common carotid artery on the B4. Um, what about the muscular group? The muscular branches, C1, C2, 3, and 4, 5, all show muscular branches. And here at the C1, you see the whole neck. The donor's head is flexed this way. And you can see the branches of the vagus nerve to the generic laryngeal muscles here. And here you can see this blue little branch is a pharyngeal branch that's going to the muscular layer behind superior cervical ganglion. This is superior laryngeal nerve that's going to be involved in airway protection. It's going right behind the superior cervical ganglion. There are these branches that go to strap muscles, like ansa cervicalis and generic laryngeal branches, as we call it. And then the recurrent laryngeal nerve goes below the clavicle and courses right below aorta. What about the cardiac branches? How do they look? They are considerably more involved than the other ones. So here you're looking at a specimen from the front. This is the left vagus nerve. A left vagus nerve comes here and branches to the superficial cardiac plexus, and then you also see the branches to the aorta, and also you're seeing deeper branches to the deep cardiac plexus. This is a posterior view. This is the right vagus nerve coming through, and it's branching to the deep cardiac plexus, and then branches into esophageal plexus. You'll see similar stuff here. Deep cardiac plexus, there's a lower descending aorta branch here, and then other aortic and cardiac plexus branches. And this image shows a full course through thorax with aorta marking the top. Slowly, this RLM branch is coursing below aorta, and then you see these branches that going to the cardiac systems, and then you see your pulmonary and esophageal branches. So this is how they look like. It's so complicated, and um, how do we map it? So in order to map it, we first have to know where they are, right? Where are these? We need to have a reference frame. So in order to have a reference frame, we selected three easy to identify, surgically identifiable anatomical landmarks. Which are carotid bifurcation, laryngeal prominence, and superior border of clavicle. And so carotid bifurcation would be here, laryngeal prominence would be here, and the superior border of the clavicle is here. And then we define some zones. Upper cervical would be the between jugular foramen where vagus nerve exits the skull and the carotid bifurcation. The mid cervical would be between the carotid bifurcation and laryngeal prominence. The lower cervical would be between the laryngeal prominence and the superior border of clavicle. Below, immediately below superior border of clavicle, we call a thoracic outlet or upper thoracic, and then below it we can call it thoracic. So we asked if this variable branch emergence be mapped onto a 1D coordinate system defined by these external anatomical landmarks a surgeon can use. Because we believe that an anatomical map becomes useful when it's translatable to OR or, a device development scenario. So how did we standardize them? So everybody's vagus nerve looks like this when you add these landmarks. And now, but no two people are the same, and neither are two vagus nerves. Some people are taller, some have longer necks, some nerves have a curved course, some form a plexus earlier. So the total length changes. There are factors related to dissection. Some people have so many branches after a certain length. It's uh, esophageal plexus forms, and it's just impossible to uh, attribute a length. It's not a 1D thing anymore, but it's a web. So total length changes a lot. And take these two nerves. These are obviously um, figures. One is long and one is short. Measure 15 centimeters from jugular foramen. On one person, it's below clavicle. On one person, it's above clavicle. Definitely two different targets from the surgical perspective. And what we did is to register or scale every nerve to the same landmark framework to make sure we can compress branch emergences from different nerves onto the same singular axis. And how did we do that? We looked at the mean distance between each of these segments. So this first segment, we look at the JF and CB for this person. We do it the same for this person. We look at the mean, which is going to be around here, and then we compress the larger one and stretch the smaller one so they come to the same length without changing the relative distance of the branches above them. And if you do it for everyone, now you have so this is a working example. We scaled each interlandmark segment to mean length of that segment of that side. Here you're seeing a bunch of different branches. This is the jugular foramen. This is from one patient, sorry, one donor with a longer nerve and one donor with a shorter nerve. And you can see different branches and how they emerge here. You can see that even the distances between their landmarks are different. So this person has the carotid bifurcation is closer to laryngeal prominence. And also, you can see that the person with the longer nerve has actually his branches to the esophageal plexus is closer to the superior border of clavicle, and it's further in the shorter nerve, but they end up being at the same place. So when you stretch and compress, you can see these kind of things, relationships better. Now it's the same length for both of them, and now we can see on this person, oh, okay, yeah, esophageal plexus branches appeared a little later, while the other branches and their relative positions didn't really change. So this was our registration framework. And So the third aim was to characterize the patterns of emergence. This is the highest level look at the our seven groups. What happens? So here, the vertical lines mark the carotid bifurcation, the laryngeal prominence, and the superior border of clavicle here. So one, two, three. And on the left, most sympathetic branches here you can see in the superior cervical ganglion. On the right, you can see it continues a little bit more toward lower. And then muscular has these two bimodal distribution, and vascular has this maybe three modes here, one, two, three. Well, muscular also has some branches here. We're going to explore this structure. When you look at here, you can see there's a lot of cardiac branches that are still above the clavicle, and think how clavicle above the clavicle portion is the more accessible both surgically and non-invasively portion of the vagus nerve. And so on both sides, on both left and right sides, there are a significant number of cardiac branches that are still above the clavicle. But we'll look at the subgroup distributions. They are different on the right versus on the left. We're going to talk about that. And then pulmonary and esophageal branches emerge the latest. And uh, so we can also ask a very practical question. Do I have this particular branch? Does everybody has all these branches? The answer is no. It's very surprising that only three of these branches were in every single subject in our cohort, which were the superior laryngeal, the recurrent laryngeal, and cardiopulmonary branches. Other than that, there were the superior cervical ganglion branches were present in 96% of people. Pharyngeal branches were present in 80% of people. And common carotid was present in 86% of people, but not all. Superficial cardiac plexus branches were present in 82% of people. Again, not all. Um, and if we go more granular and look at more granular patterns of the branch emergence and look at the subgroup level, we do see some patterns that we're going to try to quantify now. Now, if you look at here, you can see the superior cervical ganglion branches are mostly on the superior cervical region above the first vertical line being carotid bifurcation, and some sympathetic trunk sags under it. And on the muscular, these are the most important ones. You see pharyngeal and superior laryngeal almost exclusively about carotid bifurcation. There's a group that's below in the mid to low cervical, and there is another group right below the clavicle in thoracic outlet. So there's like three modes here. The same is true for the vascular too. Internal carotid artery, carotid sinus, carotid bifurcation. These are one group, look like one group. Common carotid, internal jugular vein, and general vascular look like one group. And then there's the thoracic group, which is aorta. Um, this look like plausible, and we have evidence from we have the conceptual understanding of this from human embryology that superior laryngeal nerve comes from the fourth pharyngeal arc, uh, which is a segment of six segment embryological primordial of the neck. So fourth segment in that pre-neck, and then uh, the sixth pre-neck segment is known to be where RLN emerges. So four, and the fifth one is known to be rudimentary in humans. So it's very interesting that we see here that pharyngeal and RLN, SLN and RLN in every 100% of people, and only 30% of people have these branches, which would correspond to the fifth pharyngeal arc, basically rudimentary fifth pharyngeal arc. Anyway, the same pattern here. We did show that statistically that pattern holds. There's a upper cervical group, there's a mid cervical group, and there's a lower upper thoracic group in these subgroups, and this can be statistically shown. And similar things for the thoracic branches, the cardiac, superficial cardiac plexus, the deep cardiac plexus, there's a difference between them. And on the left, so the above clavicle compartment of the cardiac branches, on the left, they go to superficial cardiac plexus. On the right, they're mostly cardiopulmonary branches. So there's an interesting difference, side difference there. And also, cardiopulmonary is more toward lower when it's on the left. And pulmonary is also toward lower when it's on the left. And there's not much difference between the emergence of pulmonary and esophageal branches. So when you go into the thoracic branch emergence and their sequence, are they like, can we really say that this distance is mostly where cardiac branches emerge and they are different than this distribution? It seems yes, we can say that cardiac branches are statistically significantly separated from the cardiopulmonary, and they are statistically significantly separated from the pulmonary branches, but not such thing can be said for pulmonary and esophageal. And also the lower the superficial and deep cardiac plexi are also separate from each other. And we also looked at the spaces where there's no branches at all. So we can, with the rationale, thinking that if we see a empty space, it would be more suitable to place an electrode so we try to also quantify branch-free intervals around these landmarks that we have detected, and we have shown that there's around a few centimeters of empty space in mean space around the landmarks, but it is very variable. And in some people, there's just no space at all, while some people have very, very lengthy no branch intervals. So, and the last part of our study was about comparing a target set of organs and branches and trying to see if we can selectively if we can turn the anatomy into a selectivity question. So let's say that I am planning to work on a stimulation paradigm and a device targeting a group of organs whose branches I want to stimulate, while another group of organs that I want to avoid stimulating. At each level, I ask two things. How much of the target pathway is still available in trunk? So let's say this is the vagus trunk, and I will place a ruler that will let me ask, okay, this centimeter, how many of my target branches emerged, and how many of my, what percentage of my target branches emerged at this point, and what percentage of the other non-target, the comparator group of branches emerged. And then I ask this for every centimeter, and I look at the difference, and then then I decide where the difference is highest is where the optimal place for me to place an electrode to target my target branches while avoiding my comparator branches. So this is what we think is very important for placing different for designing different paradigms to target different functional systems. Of course, these are inferred from the anatomy and requires more work, but this is what we think. So, selectivity obviously depends on comparison, which group you're trying to target and which group you're trying to avoid. So let's say that you want to modulate sympathetic function with your device, and you try to avoid any neck musculature related side effects. What do? You take your target group, which is going to be in our case, sympathetic trunk, and we compare it with the cervical visceral, so muscular, vascular, everything else, and we're trying to see, okay, what is the optimal place that I can place this so I can target the sympathetic and avoid these. It appears to be around 6 centimeter on the left and about 7 to 8 centimeter on the right. Um, the same, I can ask for cardiac and carotid bifurcation. So these are both cardiovascular targets, right? But we know that from the literature that the carotid sinus carotid bifurcation modulates the baroreflex mechanism, which is more related to the arterial blood pressure. And we also know that the branches that go to cardiac, to the heart, the superior and inferior cardiac branches, are mostly related to the heart rhythm and inotropy. And so we want I want to control the heart rhythm and inotropy while avoiding, and I'm trying to not lower the blood pressure. Then I need to make this comparison. Okay, so I take my cardiac branches, I compare with carotid, and where is the optimal separation? It's around 12 centimeter, and we almost get like 100% separation on the left. On the right, it's about 11, 12 centimeter too. So we can do the same for now. Let's say that I want to get cardiac and vascular. I want to have both inotropy effects and baroreflex effects, and I want to avoid muscle and sympathetic, the same thing. So I'm going to demonstrate this through our Explorer. So Vagus Nerve Explorer, this is a companion tool that we built with the paper that basically lets everyone do these explorations and see the data that was collected as part of Spark REVA program in Feinstein and with all the other collaborating labs. So we have four panels, and you have seen all of these. One panel shows the individual branches. The second panel shows the what we call subway map, the prevalences, and then you can see the distributions, and you can also look at the selectivity. Let me go over them one by one. Um, and so the individual branches here, as you can see, each row each row is a person. Sorry, each row is a vagus nerve, and the males are the blue ones. The red is the females, and at the top you see the right nerves, and at the bottom you see the left nerves. So you see here these are different. These little data points show where different branches are emerging. So sympathetic branches are the red one. Actually, let me close everything first and then start. So this is how the sympathetic branches look like in all of our patient or donors. And these are how the muscular branches are distributed. These are how the vascular branch is distributed. And these are the cardiac branches. You can see we moved below the clavicle. And this is showing these singular branches. You can also go look at a certain donor, and you can see how their branch distribution looks like. You can look at the row distances, how it was before the registration and how it looks like after the registration. And you can show all the donors, as we said, and then you can uh, show females, show males, left vagus, right vagus, any of these supergroups, like any of the groups and their subgroups too. Let's say I want to visualize the vascular ones, but I only want to have a look at the carotid sinus and bifurcation. Okay, where are they? So they're about here. Um, yeah, they're around here, most of them. Let's say, okay, I want to compare these with the aorta. Here's the aortic branches, and here's the carotid bifurcation and sinus branches. And then you can move to the prevalence view, or as we like to call it, the subway view, and you can show it. You can explore the whole data set, or you can use it as a like a presence card for each person and look if that person had a SCG branch or anything else. And then you can look at the distributions as we have looked at them, or you can make comparisons, and you can pool these comparisons. For example, you can compare, let's say, I have some pools here. I have a cardiac subtypes pool, for example, the superior cardiac plexus, the deep cardiac plexus, and the cardiopulmonary. You can pool these all three separately, and I'll show you how. You just select um, some sympathetic and muscular, and let's call it neck, and add them to your pool, and it's just going to show you the right now we're looking at the neck group, which is going to be the sympathetic and muscular branches versus the different distributions of the cardiac branches. So, and you can do this for anything. You can let's have a look at the pulmonary versus, I mean, yeah, I'm not going to do this for you. I mean, anybody that has their own study question come here and can compare different groups of them. So this is the what we see as the most important view of what we're doing. So again, the same thing. This is the length of the vagus nerve distance. So this the left would be jugular foramen, right would be the end. This would be the diaphragm, and let's see what we have here. So these are I'm going to first select a target and then select a comparator and then look at where is the high separation, functional separation, anatomical separation is. And let's say one of the things that we did there. So let's pick carotid bifurcation and carotid sinus as our target. And we're trying to, as let's pick cardiac branches as our target and and carotid bifurcation and carotid sinus as our comparator. So we want to avoid baroreflex and we want to try to modulate the inotropy and like cardiac function. So what happens then? So what it does is, so what our like selectivity engine, let's say, does is first it shows, okay, where is our comparator arising? It's going to be here. These are the sinus and bifurcation branches. And where is our target group emerging? These are the carotid branches, these are the cardiac branches that we told you about above the clavicle. And then the rest is below the clavicle. But you can see here the recruitment, like how many, what's the percentage? You can see that as I move this cursor, I can move the cursor, by the way. So as I move the cursor up, down, I'm getting less and less number of these sinus and bifurcation branches. And when I'm about 13 centimeter, almost 100% of these branches are gone now. So I can say that theoretically around the 12 centimeter of someone's vagus nerve, I shouldn't expect many carotid sinus branches anymore. So what we're saying is that then we can, this region would probably avoid baroreflex effects and would favor more cardiac effects. Uh, I think yeah, I think this is enough for demonstration. I'll go back to our presentation and oops. So yeah, and this map was built by many hands, and we're grateful to the donors and their families for because their final act of generosity made this anatomical work possible. Our paper is provisionally it's accepted in brain stimulation journal, which is we just had the news a couple days ago, and we're very happy about it. I want to thank everybody that were involved in this work and all the uh, institutions and NIH and Spark Initiative for supporting us. And I'm showing here a non-exhaustive list of people that I learned a lot from. Not everybody here was involved in this paper, but I learned from everybody here, with being the first, the utmost being my PI, my mentor Stavros Zanos, and also Mary Barb. We had a chance to work more closely during this paper, and I learned a lot from her, from Zainab, how to dissect vagus nerve, from Theo, how to reason about and think about computational aspects of the and how to quantify vagus nerve. Eric and Todd also he taught me a lot. So thank you very much. I want to thank them in front of you. So you can reach data and code, vagus nerve, Explorer code. It's on Spark Portal, it's on GitHub. You can also find the release on bioRxiv, and this is the DOI if you're interested. Thank you very much for listening.
Thank you, Siyar. That was just a tremendous opus of data. It was a true data deluge, and it's very exciting to know that there's just going to be so much information that's going to continue to come out from this study. I open the floor for questions. So if you have any questions, please drop them in the Q&A panel. We have a question to start us off, and would like to know if you have any comments on auricular or external stimulation as a result of this study.
Well, it is extremely difficult to say anything about those. Firstly, because they were so the auricular nerve, Arnold's nerve, was out of our scope. We started with superior laryngeal nerve as our as the marking at the end of nodose ganglion. So most of the branches that were a part of the neurovascular bundle there. We didn't count them, so we have no information about the auricular nerve in this.
So it sounds like future investigations for that.
Yes, probably.
Okay, so we have another question, and this question is, do you plan to relate the branching anatomy to the fascicular organization within the vagus nerve more closely? I think understanding the mapping of fascicles to peripheral branches is key for selective cervical VNS.
Absolutely, very true. I personally am not planning right now, but that's a very valid thing to speak about. And next week, I think you should join Todd's talk. Todd is going to talk about that. That's mainly what he's working on through his amazing analysis on micro CT images. So yeah, there is very important point, and there's considerable interest among the big labs to understand that, and that is exactly what is going to make this paper relevant. Otherwise, it is still a just gross anatomy, study. It needs a lot of validation. We have to look inside those branches, which fibers go there, what happens there. I also have my own another project that I'm running where I have 3D models of the entire cleared peripheral nerves, and I do see very interesting patterns there. Like U fibers making U-turns. So what are all those fibers going to do? So there's still much to learn about the branches of vagus nerve, and those work are being done with a lot of people from here, from Case Western team. So yes.
Definitely an opportunity to have everybody tune in next week to hear Todd Levy and Mabel Lynn discuss how the individual samples are being analyzed at the multiple scales. So you started at the gross dissection level, and we go to micro CT, and we go even deeper than that, looking at fascicles and individual fibers. And all of that requires an intense computational pipeline to put it all together. And we'll hear all about that next week. Same time, same place. We have one more question, and I think we have time for maybe another if one pops in. So the question is, I may have missed this, but were there significant sex differences in vagal topology? So we have left and right differences, but what about sex differences?
Yes, actually, let me find one of my let me see it. Um, oh yeah. So there are there were some side asymmetries that were uh in our well, they're not, I can't show it here. So there's not many differences between male and females. Uh, Almost everything was the same, with only exception being, I think, jugular vein. So most of the differences are like side differences related to the left side. This branch is, emerges further, and within branch like differences, but we didn't take many sex differences.
This additional question, this next question, dives into that a little bit more deeply, I think. I'm curious about how you decided to group your categories for the developmental domains. For example, ansa cervicalis is part of the muscular category, which is sorted into pharyngeal arch target related domain. But the
Yeah.
ansa cervicalis and its muscular targets are not derived from the pharyngeal arches. Additionally, any insights into upper cervical, mid, lower cervical, and upper thoracic groupings would be very helpful.
Yeah. So this is a great question. So how we So our developmental logic is based on this. First of all, I didn't define this. We have pharyngeal arc targets that these are very broad categories. We thought about it a lot. We can't say embryological neck, so that's why we call it pharyngeal arc, because sympathetic is also what we included in the pharyngeal arc branches when we made the distinction. Actually, this is going to be a more relevant one. So here, so the pharyngeal arc domain here we defined as um, any branches that go to the vascular, muscular, or sympathetic groups. And the primitive mediastinum would be the other side of things, the branches that go to long esophagus and the heart. The distinction here was the embryological ordering, where the venous pole marks the end of the neck and marks the start of the mediastinum. So these structures, we put them in those because we thought, they were related to these compartments, and about the pharyngeal arches. So yes, these structures, they're not like the ansa cervicalis is not coming from those. However, uh, uh, the pharyngeal, when you go look at the anatomy of the pharyngeal arches, you can clearly see that there's a like a vascular and then a nerve following those into these target structures, and uh, our rationale for dividing these into three, firstly here. So how did we select them? Firstly, we selected them by just looking at the distribution here initially, and we tried to see if like pharyngeal and SLN, they are branching here above the carotid bifurcation. And these two, they look like they branch from the same places. And then we did this test. And after this, we went into, okay, why could this be the case? Then we went back to, I mean, this, division during the embryological development might be the reason.
I see.
And
So
I,
let
Yeah, I think also, especially the vascular division, supports this a lot because aorta is is the lowest of those pharyngeal arteries on the left. The right side becomes rudimentary, and all the right side becomes rudimentary. The pharyngeal arteries then morph to become so the tip, the highest pharyngeal artery becomes the tip of the carotid. I think the second one becomes the internal carotid. The third one becomes the carotid trunk. And you basically see it here. So the tip of the carotid is the highest, the upper cervical, and the other branches. I mean, I definitely recommend. I couldn't really explain that a whole beautiful spatial relationship here. But if you look at the spatial relationship and look at this data, it kind of made sense. So we're talking about that.
That's spectacular. I think we have time for just one more prospective question. What single design feature would most improve on the current commercial VNS cuff based on your findings? Do you want to go out on a limb and offer any thoughts on that topic?
That's very interesting. I'm not an engineer. I'm a surgeon. But I am interested in device design and I do design things. Um, well, so we have a vagus nerve stimulator that stimulates the organs and another one that stimulates the toward the brain. And the cuff electrode it requires higher energy. That's why we use that one for epilepsy, and the bioelectronic VNS is actually way smaller form factor device with a smaller battery in it, so it doesn't need any leads, it doesn't need any batteries to be planted in somewhere else under the skin. So that would be the form factor I would go for, especially if the target is some branch that you're trying to stimulate. And obviously here we would be targeting um, the visceral system. Uh, s- I think so from a surgical perspective, above the clavicle, the cuff looks like, you know, the pretty the best option we have. But below clavicle, uh, I think designs that would let us to place things there endoscopically would be probably more important.
Okay. Well, I think this is a good stopping point because it leads right into, I think, possibly something that Todd and Mabel may touch upon in their seminar next week, which they're going to be discussing how the individual samples um, are uh, normalized and represented at the different scales using anatomical scaffolds and 3D modeling. Um, I'd like to encourage everybody to attend that seminar. Both that seminar and this one will be available on our YouTube channel. And if you go to the event page for Dr. Badaher's seminar, you'll find a link to the Vagus Nerve Atlas Explorer that he created, as well as the peer review, or sorry, the preprint manuscript, and then also links to the entire dataset collection that's available on the Spark portal. So all of the data is available for you to dive into as well. Dr. Bahadir, this was very exciting and really a tremendous amount of depth across such a incredible um, research question. So thank you for your time. Thanks to everybody else, and we'll see you next week.
Thank you very much. See you.